In this module, we're going to look at propagation from seed. The module is divided into two units, and this first one is going to look at seed structure and define some terminology. The second unit looks at the factors influencing germination and steps that we can take to optimize germination success. The learning outcomes from this unit are to define sexual propagation, describe basic seed structure, and define germination and emergence. So what is sexual propagation? Sexual reproduction involves the recombination of DNA from male and female parents and is a major source of genetic diversity in both populations and individuals within the plant kingdom as well as the animal kingdom. Plants produced from seed may look very similar to their parents, i.e. they're phenotypically similar, but they are not identical genetically. In contrast, asexual reproduction involves no recombination of DNA from male and female parents, and the resulting offspring are clones. They are genetically identical to the parents, and we'll be talking about asexual propagation techniques in other modules. In agriculture and horticulture, sexual propagation uses the product of sexual reproduction, i.e. seeds, or in the case of ferns, spores. There are several advantages to growing from seed as opposed to asexual methods such as cuttings. Firstly, growing from seed can be more economical because it's much less labor intensive. It can also require less specialized equipment, although today more and more nurseries, even smaller ones, are now investing in specialized machinery that fills plug trays with potting mix, sows seed, and after the seedlings have emerged and become established, does the grading and patching. Another advantage that seed has over vegetative propagation methods is that sometimes more seed may be available from a plant than cutting material. A good example of this is the California native sage, white sage, or Salvia apiana, which is familiar to many people as the sage that was used by Native Americans in Southern California in smudging ceremonies. This plant doesn't produce a lot of long vegetative stems, so cutting material is limited, but it does produce quite a large amount of seed. The disadvantages of seed are, firstly, that it's not suitable for hybrids, because the seed from hybrids won't come true to the parents. Hybrids have to be propagated clonally using asexual methods. Seed isn't always suitable either for non-hybrid cultivars, because the desirable physical characteristics for which the cultivar was selected in the first place may not be reproduced in subsequent generations produced from seed after meiosis has taken place. Thirdly, seed isn't suitable for crops where we have separate male and female plants and where we're wanting to reproduce a plant with a specific sex. Perhaps the male or the female plant has a desirable characteristic that we want to reproduce, or perhaps an undesirable characteristic that we want to avoid. If we're growing from seed, we won't know what the sex of these plants is until they bloom, and depending on the species, that can take up to 15 years or more sometimes. Some examples of plants where most nurseries are wanting to reproduce a specific sex are Chinese pistache, where the fruits produced by the female are considered messy by most city planners. Chinese pistache is frequently used as a street tree in a lot of cities. Fremont cottonwood, Populus fremontii, produces seed that again is considered messy by city planners, and it's also very allergenic. In ginkgo, the fruit produced by the females is really smelly, so not many people want female ginkgos in their landscape. And our California native shrub coast silk tassel has long tassel-like flowers, which give this native shrub its common name. But these tassels aren't as long and showy in the females as they are in the males. So in an ornamental situation, landscapers are looking for male plants. And finally, recreational cannabis growers usually want only females. So in all of these examples, seed is not usually the best method of propagation. Let's look at some basic terminology now. In the angiosperms, which are the flowering plants, sexual reproduction takes place in the flowers, which are highly specialized and evolved structures. The sole purpose of flowers is reproduction, and the sole purpose of showy, colorful flowers and fragrance or odors are to attract pollinators. Many plant species produce flowers that contain both male and female reproductive structures, and these flowers are referred to as bisexual, perfect, or hermaphrodite. Some species produce separate male and female flowers on the same plant, and these flowers are described as imperfect or unisexual. And plants that produce separate male and female flowers on the same plant are described as monoecious, which means one house. Mono means one or single, and ecious is derived from the Greek word for house. And examples of monoecious plants include our native white alder, Ulmus rhombifolia, willow, and hazelnut. And there are other species that produce separate male and female flowers on completely separate plants. And these plants are described as being dioecious, so two houses. And examples of plants that are dioecious are the restios, which is a family of rush-like plants native to the southern hemisphere, cannabis, kiwi fruit, conebush, leucodendron, and the other plants that we mentioned on the previous slide. Seeds are formed after successful pollination and fertilization have taken place. And these processes are covered in more detail in the Hort 1A course. But you can also find a separate optional presentation in this class's Canvas shell if you'd like a refresher on pollination and fertilization, or perhaps you're not taking Hort 1A. Seeds come in many shapes and sizes, and with many mechanisms for their dispersal. Some seeds are so small you need a microscope to see them, like the spotted coral root that's pictured here on the lower left. Other seeds are as big as an adult human's butt and are actually shaped like a human's butt, like the coco de mer, pictured on the right here in the upper left corner. Some seeds have specialized appendages that help in their dispersal. For example, the fluffy pappus of plants in the sunflower family and the helicopter-like wings on the samara of maples and ash. Other seeds have spines that cling to the fur of passing mammals. And some seeds have appendages that help them bury themselves more deeply in the ground in the wild. For example, our California native herbaceous perennial beach evening primrose has corkscrew-like appendages that help bury the seed quickly in the shifting sands of the sand dunes where this plant grows in its native habitat. Regardless of their size or shape, most seeds share a similar structure, and all seeds share similar environmental needs in order to germinate. They need water, an appropriate temperature, appropriate light levels, and oxygen, and we'll come back to these in the next unit. Some seeds, though, don't germinate even when we give them the right environmental conditions, and these seeds are said to be dormant. These seeds need additional treatments in order to break their dormancy, and we'll be discussing these in the next module. So most seeds share a common structure, and you can see diagrams of the typical structures of both monocot and dicot seeds on the right here. All seeds have a protective seed coat called the tester, which surrounds an embryonic plant that consists of a plumule, which is an embryonic shoot, and a radicle, which is an embryonic root. Most seeds, except for extremely small ones, contain an energy store of carbohydrate. In the dicots, the carbohydrates are stored in a pair of fleshy cotyledons, which are embryonic leaves, and are the first leaves to emerge when the seedling emerges above ground. Monocots have a single cotyledon, and carbohydrate is stored as endosperm. In grains like wheat, for example, the endosperm is what we grind up to make flour. The seed also, of course, contains DNA, so all the genetic material which controls the processes of germination and emergence. Let's start talking about germination with a couple of definitions and some clarification. The term germination is often used incorrectly to refer to the appearance of a young seedling above ground, and this is usually the first growth that's visible to us humans. But if we're going to be precise and accurate, germination is actually the term used to describe the physiological and physical changes up to the first visible signs of growth in a seed. These changes take place within the seed coat, so they're not visible to us, even if we're germinating the seed on a piece of moistened paper. Emergence, in contrast, refers to the appearance of the young root and the young shoot as they rupture the seed coat, and then the subsequent appearance of the shoot above ground or the substrate surface. Emergence is often referred to as germination because this is the first growth that we see. In some plants, the young root sometimes emerges and establishes itself several days or even weeks in advance of the shoot. An example of this is the coast live oak, Quercus agrifolia, and this growth habit is an adaptation to survival. It enables the root to become established and tap into water sources so that the seedling has a better chance of survival in the wild. Let's summarize what we've covered so far in this unit. Seed is produced as the result of sexual reproduction in plants, which involves the recombination of DNA from male and female donors. Male and female reproductive structures can be contained within the same flower, or in separate flowers on the same plant, or in separate flowers on separate plants. Seeds come in many shapes and sizes, but all share a similar structure, consisting of a seed coat which surrounds an embryonic plant and carbohydrate reserves. Germination refers to the activation by water of enzymes within the seed that start the germination process. Emergence refers to the appearance of the young root and the young shoot as they rupture the seed coat. Now that you have an understanding of the basic structure of mature seed, take this short quiz to check your understanding of this material, then head over to the second unit in this module to learn about the factors that affect germination and the steps that we can take to optimize germination success.